I'm very pleased to welcome all of you here. So we have here in the audience quite a few people, but I think even more online. So I'm really curious to hear also where you're all coming from. So please use the chat function to type in the country where you're coming from, so we have a bit of a visibility who is attending. Here in the audience, do we have people from Essen? I can see 3 hands. Any other cities? What is the other city? Würzburg, great, welcome. Anyone I missed? Okay, so we have Essen and Würzburg here. And let's see, who do we have in the audience? Also another note, please note the session is recorded. The reason is also there are some people that cannot attend today. So we want to make sure that they can have visibility on the presentations today. So that's the note that I have to say up front. But then I'm very excited to kickstart the event. So today we are actually here because Essen, or more precisely the working group of Translational Tumor Research, is now a qualified service provider for HCG technology. And with that, I would like to hand over the official certificate here to Dr. Myringer. So congratulations to you, and in particular also your team that did a great job in performing the analysis in a very efficient way so that we can really trust you're doing the analysis in a way that we would expect. So thank you so much.
Thank you very much.
So let's take a look at the agenda. I did already dive into the welcome and opening. So Bernd, can you share what nationalities we have attending? Just a second, we need my...
شكرا لمساهمتك يا لولو. خلينا هلا بعد شوي حنروح نشوف ليس and then بتضحك ماما كانت حكيت مع خالد معي.
Thank you. So for those that are not speaking, please keep your microphone on mute. After each session, we will have a Q&A, so you can ask questions. That's the time where you can activate your microphone. And also, if in case you prefer to use the chat function, that's also an option to simply type in the chat your question, and we can then address it at the end of each session. So let's take a look at the agenda. We will have Dr. Myringer present his working group, and then I will provide a short introduction into HCG molecular. We will then have 2 scientific presentations from Dr. Borchett and Ms. Giallini, one more on the FFPE side of things in translational tumor research, and the other one more on the microRNA side and CSF analysis, and we will then end with a closing remark. So I would like to introduce Dr. Fabian Myringer. He is the group leader of the working group Translational Tumor Research here at the Institute of Pathology, University Hospital Essen in Germany. Obviously, he's supervising students with their thesis. Also, he's a lecturer in molecular pathology and tumor biology. Quite interesting, I found to discover that he has a bachelor in biotechnology, but then very likely decided he wants to do something more patient-oriented and studied medicine. And here also at the University Duisburg Essen, where he also did his assistant professorship in molecular pathology. After then also spending some time at Charité Berlin, he now is fully established with his working group here in Essen. Dr. Myringer, please.
Yeah, thank you very much. Maybe we can turn on the light a little bit for here in the room. No? I don't know. Okay, I think maybe it's a little bit better now. Yeah, so today. Thank you, first of all, for the nice introduction, and thank you for your interest here and the opportunity to present my research group here, also online. These are my closing remarks. As mentioned before, I'm heading the Translational Cancer Research Group here in Essen at the Institute of Pathology. We have a long time experience, more than 10 years now, with digital gene expression analysis. I think this is the main focus we are performing here. Already 2010, we got the nCounter Flex system for digital gene expression analysis. And 2014, then we get the upgrade of the machine for diagnostic purposes and become 1 year later official Prosigna test center for the prognostic breast cancer gene signature assays. Then 1 year later, we installed an Affymetrix microarray system. We extended our technology, therefore, to now make whole transcriptomes to go more on the depth of the different cellular processes. And 2020, then we get an upgrade of our, or an amendment of our Flex system by the GeoMix digital spatial analyzer to now analyze the different cell types, the specific cell types in the mixture, in the complexity of tumors. And now last year, in the middle of last year, we captured the HGG HSEC system from another laboratory from pathology and integrated it now here in our workflow. I think this is a very, very nice extended to our technology we have. It's perfectly in the focus. So we now also can make whole transcriptomes and broad transcriptomic analysis from even very small samples. Let's say a biopsy with only 1 millimeter of tumor, we now can make a whole transcriptome without RNA isolation, as Sabrina will talk about later. Maybe a few words to my research group. So I'm the group leader here. I have 2 postdocs, Michael Wessely and Sabrina Borchett. Sabrina is making the administrative stuff, is taking care of all the ongoing works and all the daily bread and butter we have. Then Michael is our bioinformatician, so we have our own bioinformatician in the group. Then I have 3 technicians, Yvonne, who is managing the lab, Sophia and Moni, and some postgraduate students. They're all MD students at the moment with experimental MD thesis, and a few students, bachelor, master, who have some turnaround in our group. For sure, our main mission we have is cancer research. So I'm coming from the cancer research. I really like cancer research very much, but we are open-minded for everything. We have interest in nearly everything. We have corporations for, I have some examples here, neurological diseases. We have corporations for cardiovascular diseases. Also, we have to do a lot with other research groups which make asthma and COPD. We have some projects that are not entity-driven, but some kind of signaling by microRNAs and extracellular vesicles and so on. So we're very open-minded. We are interested in nearly everything, and also very eager to see new insights, to see other biological functions, other mechanisms, other diseases like before. Besides these corporations, we also provide just service. So we have a lot of partners that don't want to do a real cooperation. They just want to get the data. And therefore, we also provide services like a core facility. We are certified core lab for the nanostring nCounter system, certified core lab for the GeoMix DSP system. We are official test center for the gene signature arrays out there. And we are now, since end of last year, a certified service partner for the HGG HSEC system, which we also can provide here. I think what we do from our own research is mostly classic translational cancer research. So we are focusing on the one hand, on stratification of patients, go away from this one-size-fits-all approach to the more stratified approaches for classic therapies, targeted therapies, or immune therapies nowadays. But on the other side, we are also very interested in the complexity of tumor, in the interaction between the different components, comparts, different cell types within these tumors. So we're looking what are the activating mechanisms, what is it, what activates the tumor cells, what properties they should have, and under what selection pressures these tumor cells with these new properties can grow, can enhance in the tumor, and therefore get, for example, an immunologically resistant tumor, some kind of immune escape, but also other selection pressures we want to analyze. And on the other hand, I think the newer projects we have, the nowadays things, are more the other compartments, not the tumor cells itself. For example, fibroblasts or other cells interdispersed, immune cells in the tumor, how they activate the tumor, how they interact with the tumor, how they communicate, and under other communications, pressure, selection pressures, how is the activation of those other compartments by the tumor cells, and what factors, what signals they have to release immunosuppressive factors, get an immunosuppressive environment, tumor prognosis, tumor progression, things like this. So these are the main areas of focus we are doing. I think beside a very nice expertise in gene expression analysis and a very nice expertise in transcriptomic analysis, we provide more or less all a very nice insight in statistics and bioinformatics. So for the normal stuff, I think we are all firm to do it. Something like differential expression analysis, something like Geneset enrichment, pathway analysis, and so on. And for the more deep things like deep learning, like machine learning based tools, like prediction models, we have our own bioinformatician who can provide this expertise. And I think in the last time, we mostly make some kind of interaction networks, interaction between different cell types, and how the complexity of a model such a tumor is working. So these are more the basic things we do. We are translational cancer research, and I think really that we want to do this translational part, bring this information from the basic to the clinics, and therefore we make a lot of single marker, multi-marker models, try to get either weighted linear sums or binarized models or whatever to get scores to predict the outcome of the diseases, to predict the response to therapy. Then we try to model different models based on these scores we are generating. And in the end, we are providing some kind of software solutions, tools for the clinicians, where they, for example, can type in the gene expression here, and then they get a prediction here for platinum resistance, the chance of platinum resistance in ovarian cancer, or survival predictors. Or we also make some diagnostic tools, software solutions, where, for example, here, the present samples then can be distributed between IgG4, ophthalmological inflammation from lymphomas, so that the clinicians really get a helpful tool in their clinical daily management of the patients and to transfer this information here. So I think this is our mission. This is what we want to do. We are interested in everything, so you can come with every kind of project, every kind of idea or cooperation. We are open-minded for everything, and this is what we can provide. I think we have a very nice technological part, very nice statistics and bioinformatics behind, but we're all aware that just an association, a correlation we find, not ever reflects a causal association, a causal combination between the different things like here. But I think still that with the new technologies, we have since last year, we really can provide more and deeper insight in this complexity of diseases like we understand it nowadays. And for you, I think you're also aware, looking outside, that there's nothing like free lunch in life. With this, I really want to thank you for your attention and for the opportunity to present it, that you're all coming here, and would be happy for any questions or cooperations or project discussions in the end. Thank you very much.
Thank you, Dr. Myringer. Are there any questions? So if you're attending online and you have any question, function of Teams or simply type a question in the chat or unmute yourself to ask. Okay, looks like currently there is no questions, but maybe they will come up later. So we proceed. So I will introduce HGG Molecular and what we do. So you did hear Dr. Myringer speak about multi-markers, and that's where we are heading. The more information, the better. And where HGG Molecular positions itself is really on the RNA. There is really great value in all the levels of biomarkers that you can assess. So DNA is the blueprint of life. So obviously, if you find any biomarker based on DNA, that's really informative. And in particular in cancer, it's the main driver of many diseases. But even in lung cancer, which is a very common disease, you still have 20–30% of non-small cell lung cancer without a mutation. But not even speaking about other diseases where mutations are not the driver of the disease, but you really need to understand the different regulations that are playing there that can drive the disease. And that's really where RNA can help. First of all, you can assess the mode of action and the driver of the disease right on the signaling side of things. But second, you can also multiplex. You can look at many markers at the same time. And this is, on the other hand, a bit more challenging if you're looking on the protein. Obviously, protein is the executor of the signal, so it's the gold standard to assess. But often protein is more limited to assess, or even, as I mentioned, when you want to look at many markers at the same time, that can be challenging. But also in terms of the folding of the protein, sometimes it's simply really hard to have a reliable technology to detect it. So that's where we come in for RNA analysis. And what is the gold standard today is really RNA-seq. So we are kind of an RNA-seq approach, but we simply simplify it. So the NGS is the readout that we are using, and you can look here at the graph and see it's much shorter and simplified. And many of the steps are avoided or optimized. So when I speak about avoidance, it is an extraction-free approach. So no RNA needs to be extracted. Obviously, that reduces a lot of biases that are associated with that, and also reduces cost and labor of these steps associated. But also, even more importantly, you have low expressed markers that can be lost while you're extracting, and this is what is captured with our technology. So here in this slide, you can see really the key advantages of the EDGE-seq workflow. First of all, the sample preparation is straightforward. It's a simple lysate that you generate from many different sample types that we can work with. And it also allows you to work with very little material. So we have a low sample input protocol. The next step is then the library preparation, which is mainly optimized by our EDGE-seq system. And you can here run it without the extraction. So it's hybridization-based chemistry where we have the quantitative nuclease protection assay automated, and that integrates then in the standard NGS workflow of the Illumina or Thermo Fisher sequencer readout. The next highly facilitated approach is data analysis. Obviously here in Essen, we have great experts, so if you want to dive deep, that's also a resource that is available for you. But if it's a first QC analysis or first differential expression analysis you're interested in, we have a great tool that you can also use for our customers free of charge that really simplifies the complex analysis of the gene expression data that you're generating. Speaking about low sample input, we often hear from pathologists tissue is the issue, so you never have enough, but even in other sample types, like we will hear also Adriana speak about CSF from preterm infants, so obviously you want to take as little as possible. So HG really has a great advantage that you use very little material. You have the validated sample types listed here, but we can even go beyond that. So there are publications out there on urine, on exosomes, on extracellular vesicles, CSF you will hear, and many, many more. But speaking here mainly in the pathology focus, FFPE is a real challenge for gene expression analysis. And in particular, archived old blocks, we have highly degraded RNA, and many technologies fail to either extract even sufficient material of RNA or sufficient good quality of RNA. And then even there's a dropout in what is actually really analyzed. But as our chemistry is quite straightforward with also highly degraded material, that's where we have really a great advantage. And the amount we are using is a single section of 6 to 11 square millimeter. So depending on the size of the biopsy, it can be 1 or 2 sections. Plasma serum is mainly used for microRNAs. So here we use 15 microliter, but also you can do EV analysis or exosomes. And then obviously you have to have a protocol where the volume is a bit larger. PAXgene is also a standard sample type for mRNA analysis because it preserves the RNA. And here we work with 400 to 500 microliter, depending on the assay that you're running. Cells, 3,000 cells. And you can see there's extracted RNA listed. So if you have that on stock, we can work with it, but we prefer to work from the crude sample whenever possible. So why extraction free? It's quite innovative. That's why I would like to highlight. what the approach is. So you can see here in the pictures, how we do it is simply we assess the size of the material that we are using based on the H&E. And then, so this is the example for FFPE, we scrape off from an unstained slide the area of interest and simply put it into lysis buffer. That's it. You can incubate and then it goes into our system. So quite straightforward. But what is so interesting about extraction-free is usually when you extract the RNA, you have specific length of fragments that you are enriching. So if it is an FFPE extraction kit, it's 70 to 100 nucleotides and above. If it's a standard kit, it's only a 200 and above. So it means if you have fragments that are shorter because of a high degree of degradation, you will not detect them. So if you're now interested in identifying biomarkers, developing signatures, and you are missing these shorter fragments, also because maybe they're affected by low expression and they are not enriched by the extraction, which we know is true for the low expressed markers, you're actually not detecting your marker of interest that can really drive your biology, but also be a relevant marker for therapy. So that's why an extraction-free approach is very helpful, and we are using these so-called QNPA probes that have 50 base pairs. So it means anything that has 50 base pairs will be detected. Also, what you need to keep in mind, there is a loss. I mean, the more you manipulate your sample, the more material will be lost. So the gold standard kit provider assumed 20 to 30% loss. This is just here a short graph on our service lab. We have tested various sample types, and you can see here the performance. So not even speaking on comparison to RNA-seq, where you have 15 to 20% of the samples not generating high enough RIN or DV200 values. And even for the remaining 80% of samples, you still have a dropout of the samples that actually generate some data. So what you can see here, even with bone marrow FFPE biopsies that we have run, FFPE is the most challenging sample types. We have a pass rate of 90%, so really great performance. The chemistry behind the quantitative nucleus protection assay is simply a very robust chemistry. We have not developed it, it has been out there, and we simply optimized it. And how it works is we add protection probes specific for the sequence of interest. It hybridizes in the lysate, and what we actually capture is then the protection probe that did hybridize to a target sequence. We do that by simply adding sequencing barcodes and adapter in a PCR run, and we can then quantify and pull the samples and load it onto a next generation sequencer. So what comes of the and the sequencer is done in the FASTQ file. We put it onto our parsing software that generates this raw data file in an Excel format where you have the raw counts, simply the number of sequences that were read for a specific gene. You can work with this, or you can use our Reveal software for first analysis on the quality controls, so which samples did pass the quality metrics. You can do some normalizations in there, but even here directly extract publication quality figures that you can directly import in your manuscript or lab meeting or anything like that. In terms of assays, the transcriptome and Melanoma panels are the most widely used. We have a more focused panels as well, but since the transcriptome panel has been launched, this is really the key advantage, because we can look almost at the whole transcriptome with close to 20,000 transcripts from a single FFPE section. So it's always better, as we heard, multiple data points and integrating it, and then filtering the bioinformatics side for your key drivers. But the beauty is even you can run both microRNA and mRNA on the same sample. You don't have to rebiopsy or even take another sample. You can simply generate one lysate and run both assays on that. And with that, I would like to summarize. The beauty of HEG is basically you get more data from less material. You have very robust data, even on highly degraded material, such as archival FFPE, and you can work on many different sample types. You have a very fast turnaround time with an integrated bioinformatic pipeline, and you have strong agreement still with traditional RNA-seq. The beauty is also it's a really diagnostic grade assay, so you can run from discovery down to a diagnostic assay on one technology. With that, I'm at the end of my presentation, so are there any questions? We have a question here in the audience. Just a second while we're running over the microphone.
Thank you for a nice overview. My question would be, concerning other techniques like nanostring. What would you say is the standalone criteria concerning technique, quality, and also data? So, if I understood correctly, you can also do whole transcriptome. So this would be probably one standalone criteria. But are there any others?
Yeah, very good question. So as you already picked, it's the number of genes where nanostring is limited with 800 genes, and we can go up to the whole transcriptome, so meaning 20,000 in comparison to 800. That's one point. The other point is the extraction free. So nanostring usually needs to extract. I think for some samples, like the very good quality ones, they have. Yeah, so I think it depends a bit on the sample type where they can do it extraction free, but we do it as a standard. Anything we prefer actually extraction free. And then I think the number of samples they can work, it's quite similar. They also have their bioinformatics suite, so I don't have insight into how straightforward it is, so I cannot speak about that. And the beauty here is basically you have also the dynamic range of NGS for HCG. Nanostring has their own barcode-based readout, which has a bit of a different sensitivity and dynamic range.
And concerning fragmentation level and there are different fragmentation level higher HCG and
So nanostring has also that great advantage. It works really well on FFPE. We have done a number of comparison studies where we have seen a little bit higher dropout on nanostring side versus HCG, but in general, nanostring is still better than RNA-seq. Thank you. Any additional questions from the online audience or even here in the auditorium? Okay. If there's questions coming up, we can also then have time at the end. But with that, I would like to then introduce Dr. Sabrina Borchett. She's here from the working group of Dr. Myringer. She's a postdoc in his group, and she's also doing the scientific management of the working group. And she's also involved in molecular diagnostics, so doing quite a lot of work. She has a bachelor in biology and a master in medical biology, and a PhD also done here in the working group of translational tumor research at the Institute of Pathology at the University of Duisburg and Essen. So Dr. Borchett will speak about nuclease protection-based gene expression profiling and its application in translational tumor research. Dr. Borchett.
Yeah, thank you very much. And hello, I am very pleased to present to you today two of our projects we have performed with the HTG-HSEQ technology. This is my disclosure information. And yeah, this is a short overview of the two studies. The first study was a feasibility study of the HTG transcriptome panel, which consists of about 20,000 targets, and we already published this study in last year in September in the journal Cancers, and there we analyzed synovial sarcomas and its morphologic mimicas, spindle cell sarcomas. And in the second study, we analyzed primary uveal melanoma samples with their respective metastases, with the help of the oncology biomarker panel, which consists of about 2,500 targets. The background of the first study is synovial sarcomas represent a group of soft tissue sarcomas which affect a wide age range of patients, so it includes adolescents and young adults. and it's mostly a very clinically aggressive tumor. Many patients suffer from local relapse or distant metastasis, and even after more than 20 years, it's possible that late progression can occur. And synovial sarcoma is a paradigm for gene fusion-derived, gene fusion-driven cancer because it has it's characterized by a gene fusion of the SS18 gene together with SSX1 or SSX2, and in rare cases SSX4. And the spindle cell sarcomas are morphologically similar to synovial sarcoma, but they have a clinically different behavior, but they are thus called the morphologic mimickers, and these exhibit no gene fusion of the chromosome X and 18. The aim of our study was to compare these two entities, but the main part of the study was to test also the robustness of the HTP panel, because we already have results from the routinely diagnostic of our samples. We have information about immunohistochemistry, fluorescence in situ hybridization, and targeted RNA sequencing. And yeah, we wanted to validate these results with the HTP panel. And the results, we analyzed the results with the help of the Reveal Analysis platform by HTG. This is a web-based analysis tool, and this is the welcome platform of the website. And first of all, we have to make a quality control of our HTP run. So there are 4 QC metrics. The QC0 gives us information if we have sufficient RNA in our samples. The QC1 metric gives us information about if we have sufficient read depth in the sequencing run. And the QC2 metric gives a failure if there is a too high background signal. And the QC3 gives us a failure if there was an incomplete digestion of the genomic DNA by the DNAs. And these are the plots of our run. The QC0 plot we see here, each sample is under the threshold line. And so we can say each sample has passed. So we have sufficient RNA in each sample. The QC1 plots, there also each sample has passed, so we have sufficient read depth. And the QC2 plot shows us we have no too high background signal, so there are a few samples showing a higher signal, but it's on the threshold still. And the QC3 metric shows us that we have a complete digestion of genomic DNA. And in the principal component analysis, we can see that the gene expression pattern of the spindle cell sarcoma differs from that of the synovial sarcomas that are highlighted in yellow dots. And also the volcano plot shows us of about 6,000 genes showing significantly differential expression. And here we see the top 10 upregulated genes in synovial sarcoma and the top 10 upregulated genes in spindle cell sarcoma. And it's very interesting but also very important to see that the TLE1 gene was is under the top 10 upregulated genes in synovial sarcoma because it's an already established marker in the routine diagnostic. So we have for example, immunohistochemistry staining of the spindle cell sarcomas, which are completely negative for TLE1 staining, and the synovial sarcoma are clearly positive for TLE1 staining. And it's very interesting that the protein expression correlates with the RNA expression we detected with the help of the HTP panel. And our conclusion of the study is the HTP panel is a feasible method for gene expression analysis in a large scale It revealed robust results, and it's a robust method for the search for biomarkers, especially in limited tumor material. So we are talking about FFPE material, and even in biopsy material, we can get robust results of these samples. And yeah, we could prove the already identified overexpression of the of TLE1. And now I come to the next project about uveal melanoma. So melanoma is a tumor that arises from melanocytes and which are located at various anatomic locations. And among ocular melanomas, 83% arises from the uvea. So this encompasses the ciliary body and the choroid and the iris. And uveal melanoma is divided into two molecular classes of predicting metastatic death. So the class 1 uveal melanoma show a low metastatic risk, and they exhibit a disomy of chromosome 3, while the class 2 uveal melanomas have a high metastatic risk with, and these show a monosomy of chromosome 3. And uveal melanoma often metastasizes into the liver with 88%, but it could also metastasize into the lungs, into the brain, to the lymph node, and other sites. And with our project, we were interested to find or to look if there is a molecularly predefined expression pattern that can predict the localization of the metastasis. And therefore, we had 14 tumor samples of primary uveal melanoma and 10 respective metastases. 8 were from the liver, 1 of the lymph node, and 1 of the humerus, so the bone. We had 1 patient that didn't show any metastasis, and from 3 patients, we only have the primary tumor sample, but we have the information about the metastasis localization, which are the skin, the brain, or the liver. And the first part of our analysis was to find differences in gene expression in primary tumor samples. And in the second part, we wanted to find differences in gene expression in metastasis only. And in the third part, we wanted to analyze if there is a change between primary tumor and metastasis. And in the fourth part, we compared the primary tumors with their respective metastases. And the analysis of the primary tumor revealed 2 distinct gene expression patterns, two groups with distinct gene expression pattern, highlighted by the yellow lines here, please. And also the hierarchical plot shows these both groups. We name it A and B. And as you can see, there is no correlation to the localization of the metastasis. So here the gene expression of the primary tumor is shown, but the metastasis sites are not clustered. So we cannot say that there is a predefined pattern of gene expression which can predict the localization of later metastasis. Yeah, then we performed the Geneset Enrichment analysis. This is a computational method to identify classes of genes that are overrepresented in a large set of genes that can be associated with a phenotype. And our 2 groups are shown here. So the one of the group showed a rather immunogenic phenotype with pathways being affected like autoimmune thyroid disease, primary immunodeficiency, antigen processing and presentation, and so on, while the other group rather tends to be a desmoplastic phenotype, because here are pathways affected like TGF-beta signaling pathway, cell cycle, DNA replication, and ECM receptor interaction. And also the volcano plot shows on the left side that there are differentially expressed genes that are related to the immune system, like HLA-A, HLA-B, and the immune T-cell exhaustion marker LEG3. But also FGL2 is assumed to promote tumor progression by inhibiting the dendritic cell differentiation. And now we come to only metastasis, the analysis of these. There we also find two groups of that showed a distinct gene expression pattern. The liver metastases are evenly distributed in these both groups, and the lymph node metastasis is excluded from these both groups because they show this metastasis showed a completely different gene expression pattern. So also here a hierarchical plot, and we see we excluded the lymph node from the further analysis. And also here we performed a Geneset enrichment analysis, and we also found here again an immunogenic phenotype with pathways being affected like pertussis, amoebiasis, antigen processing and presentation, lysosome, phagosome, and malaria, while the other group showed a clearly liver metabolism-related phenotype with pathways being affected like drug metabolism, biosecretion, metabolism of xenobiotics by cytochrome P450, and glucagon signaling pathway, and so on. And by the analysis of here, we wanted to find the change between primary tumors and metastasis, but as you can see, here's no correlation, so there's not a real change to see in no clustering. And on our fourth part of the analysis, we wanted to compare the gene expression of the tumors, of the primary tumors with its respective metastases. And we found here significantly differentially expressed genes. And it's interesting to see that of those genes, these are in the primary tumor, these are expressed in the primary tumor, but get lost in the metastasis. And with this plot, we wanted to analyze if the immunogenic phenotype of the primary tumor is still the immunogenic phenotype of the metastasis, because especially those might benefit from immunotherapy. And interesting for us is the blue bar. So these are really both the immunogenic phenotype, but also from the desmoplastic phenotype of the primary tumor, there's a switch to the immunogenic phenotype, which also could be interesting for immunotherapy. And so I come to the conclusion. Gene expression pattern of primary tumors does not indicate where the tumor metastasizes to. We found completely different biological groups with distinct phenotypes. And in both primary tumors and metastasis, we found an immunogenic phenotype, which could benefit from immunotherapy. And currently, we are analyzing the clinical data of our cohort, and 50% of these patients were treated with immunotherapy. And it seems that the patients with this phenotype, with this immunogenic phenotype, really show enhanced response to immunotherapy. But the cohort is relatively small, so we cannot give a real statement about this, so we have to further analyze this. Yeah, and the last point is the lymph node metastasis that showed a completely different gene expression pattern, which could be due to biological adaptation or contamination of lymphocytes also. Yeah, and then I would like to thank you. Thank you very much. And if you have questions, I'm happy to answer it.
Thank you, Dr. Borchett. Really great presentations. So are there any questions from the audience? Very well, everybody's picking their brain. One question from my side. I think it's really interesting to see how we have this switch from the primary tumor to the metastasis. I'm planning to also dive deeper, maybe also in a blood analysis on extracellular vesicles to see if we can find some drivers towards metastasis that also can educate that.
It would be very, very interesting for vesicles, to analyze the vesicles. So we have not the methodology, but it would be that's a good idea to maybe in cooperation with other groups we can isolate for the analysis.
Thank you. Any questions? Okay, there seem to be no further questions, so you were very clear in your presentation. Thank you so much. With that, we now come to the next scientific presentation. We're shifting gears. We are moving away from oncology towards neurology and moving away from FFP as a sample or a tumor sample to CSF. And we want to look for microRNAs in particular. And I'm very pleased to have Andriana presenting here today. As she is at the end of her PhD thesis, very close to submission, she can unfortunately not be here today. So she's connected virtually. And while I'm introducing you, Andriana, you can start sharing your screen, please. Okay. So Andriana Gialili is working in the group of Professor Axel Heep in the prenatal neurology at the University of Oldenburg. And this is a PhD program in collaboration with the University of Bristol. Andriana did her bachelor in biology at the National Kapodistrian University of Athens, and she did her master's in molecular neuroscience at the University of Bristol. And today she is a PhD candidate in neurobiology at the University of Oldenburg in collaboration with the University of Bristol. And today she is in Bristol, I believe. Andriana, can you hear us? We can already see your screen. So we can see if we can hear you now.
Nice, thank you very much for, can you hear me?
Yes, perfect, thank you.
Great. Thank you very much for the invitation and for the opportunity to share our project and how ATG incorporate to our project. Our project is in microRNA profiling of cerebrospinal fluid from preterm infants following perinatal brain injury. A little bit of introduction. So preterm infants are at high risk to develop a brain injury during the first hours of their life, and it is injury called intraventricular hemorrhage. Babies born in a really low gestational age, less than 29 weeks, and in a really low birth weight, are at high risk to develop this hemorrhage. This hemorrhage occurs at the area which is called germinal matrix. I am not sure if you can see my pointer. This area is the walls of the ventricles, and after the hemorrhage, blood fills the brain ventricles and the cerebrospinal fluid CSF that runs the brain ventricles, and blood clot blocks the CSF reabsorption, causing the accumulation of the CSF inside the ventricles. There are many reasons that cause this injury, although the fragile vascular rate of the area at this developmental age and the disturbances in the cerebral blood flow are among them. At the moment, clinicians usually diagnose the injury by ultrasound, and the outcomes of the disease can vary. So these infants usually have so cerebral palsy, cognitive deficiency, learning difficulties, and many different multiple disabilities that, of course, affects the life of the same people, but the families, the health system, and the society in general. So it's quite important to study this disease. At the moment, there is no an active treatment to prevent for this neurodevelopment outcome of the disease, but neither to prevent for the injury to occur. But the clinicians manage the disease by using external ventricular drainer device, and they drain the extracellular, the CSF, to both clean the CSF from blood and to minimize the pressure from the accumulation of the CSF. This CSF at the moment is a clinical waste of a routine of a routine procedure, and as we thought that this could be a useful source for information that could depict what is happening inside the brain, and why we thought that the CSF can depict what is going on, the ongoing situation of the brain after IVH. CSF is the fluid that is produced by a choroid plexus, which is the tissue that perturbates inside the ventricles and creates the CSF blood barrier. It is known that the CSF, when it runs through the brain ventricles, carries signal molecules that they are both secreted and also will be absorbed by the cells that they are in the surrounding tissues where the CSF run. And that's why CSF participates both in cell communication, but also can depict what is happening in the brain at this time point, at every time point. At the time point that the intraventricular IVH occurs, the main cell that surround the brain ventricles are neuronal progenitor cells, NPCs, which are the cells that will give rise to the whole new brain cells. There is brain cell types that they will create the brain during development. Although there are many questions about how the CSF composition changes after IVH and how these changes can affect the surrounding NPCs and the surrounding tissue. So the aims of our project was to study the signaling molecules that CSF has after IVH and to unravel the interaction between CSF cues and the surrounding NPCs after IVH in the developing brain. For all the different factors that there are in the CSF, we thought to focus in microRNAs, which is there are small non-coding RNAs that after their biogenesis, they go and bind to the mRNA target, and they cause either the inhibition of those mRNA translation or microRNA degradation. Although the microRNA can also be enclosed in small membrane vesicles and extracted to the extracellular space, like the cerebrospinal fluid, and then they can be uptaken from a different recipient cell, and then they are released in the recipient cell, and they can go again and find their mRNA target there, and they can change the phenotype of the recipient cell. So the microRNAs participate in cell communication, and also because it is known that there are alterations of the mRNA expression in different diseases. They have been suggested as potential biomarkers or as potential non-invasive therapeutics. So we thought it is an important molecules that we could study in the CSF from IVH. First, we started with a small pilot experiment project where we wanted to test if the CSF from IVH patients have these small extracellular vesicles. that they contain microRNAs, and we did electron microscopy, and we identified these circular small vesicles that they have the size of the extracellular vesicles, and also stain them for markers for these extracellular vesicles. And then we study the microRNA content of those extracellular vesicles. At that point, we picked 5 brain and CSF enriched to test if they are included in these extracellular vesicles extracted from the CSF of preterm infants. And we saw that these microRNAs were expressed, and also their expression was changing over time, over treatment progression in samples taken at different time points over treatment progression. So since we saw that this CSF contains extracellular vesicles, and also the microRNA content of this CSF changes over time, we wanted to go bigger and do a whole microRNA transcriptomic analysis in our CSF samples. But the issues that we had, it was, as you can imagine, the volumes of the samples that we have are really small. And the RNA yield that we have from these are really low. So that's why we thought that the ATG technology might suit to our project, because it requires a really small volume that we could provide, and also we didn't have to do the RNA extraction step that for us it was quite difficult, and it was putting us a lot of limits. So that's why we concluded to do this, to use this technology. And now we'll give you a quick project overview, as our findings are about to be submitted for publication. So to understand what actually this next generation sequencing offers to our project, we use samples that were extracted between the 1 and day 30 after birth. And this, so after they will call early CSF, and CSF samples that were uptaken between 60 and 100 days after birth, and these will be called late CSF samples. And we want to do the analysis to see if the microRNA content of the CSF changing over time. So that's why we did we used the whole microRNA transcriptome assay from ATG. It screens from a bit more than 2,000 microRNAs, and likely all our samples pass the quality controls that they have been explained from previous speakers. So then after that, we use the Reveal software from ATG to do a PCA analysis of our next generation sequencing. These 2 samples were extracted from our analysis because the one was identified that the patient was not meeting the inclusion criteria because it had a congenital disease and not intraventricular hemorrhage, and the other was outlier, and the patient was coming from a different hospital, so we couldn't record the whole history of this patient. As far as the rest samples, we saw that all our late samples were in the same creating a group, while the early samples were in creating a separate group, a bit more diverse though. So this shows that the samples, the microRNA content of our CSF changes over time. And then we did we just wanted to see which ones are significant differentiate microRNAs. And here is the volcano plot that saw that approximately a bit more than 500 microRNAs were differentially expressed in late samples compared to our early samples. And that was quite interesting. And then we did a gene ontology analysis with different microRNA tools. And this analysis shows us the different pathways that they are involved in reactive gliosis, which is the reaction of the brain in central nervous system injury. So this was all the bioinformatic analysis that we did. And then we went to validate this analysis in vitro, and we use human fetal neuronal progenitor cells as the cells that are around the ventricles at this time point in the brain is progenitor cells. and we treat them with CSF from our patients. And then we actually study the pathways that our microRNA analysis had revealed. So at the conclusion, we saw that actually studying microRNAs in the CSF was quite useful because it was able to reveal signaling pathways that are involved in reactive gliosis seen in the brain of those babies. And maybe of those pathways could be potentially used as treatment targets after further investigation. So that was a bit of an overview of our project, but of course, all this not one person. I also have my supervisors from older groups, like Professor Axel Hipp, who is also in the audience, and also from our collaborators in the University of Bristol, ATG, and of course the patients and their families that they donate their samples. Thank you very much. Any questions? I'm happy to answer.
Thank you so much, Adriana. Really wonderful presentation and the insights that you generated on these valuable samples. So are there any questions? I start with one question. Very interesting that you then also did the transfection of the cell culture with the CSF. So in order to validate also if the let's say the interaction of the microRNA with the target mRNA is also validated in the cell culture, did you plan or have you already done the mRNA analysis on the cell culture that is then affected by the microRNA's other signaling in the CSF?
No, we haven't done the microRNA analysis in the cells. We only focus at the moment on the pathways that they were being seen revealed by our microRNA analysis. So we were only targeting like the proteins that we think and the targets, the signaling pathways. Okay. But it would be actually a good suggestion to see the microRNA content of the cells as well.
Mm-hmm. And just one more question, as you were saying in the microscope that you have EVs, but the analysis was the whole CSF. So how much do you think is coming from the EVs and how much is circulating?
Yeah, that's a good question. We started with a whole microRNA analysis because it was our kind of first attempt to see what we were going to get because we didn't know. So if we were doing the exosome, it would be limiting again the RNA volume that we had. And I think it will be good to go further and check the exosome content as well. And I hope, I guess most of it will be coming from that. Although microRNAs are in the fluids, could be different, like RNA binding proteins or in different ways, but probably most of it comes from the exosome, I would say. But this needs to be tested in further.
Okay, thank you so much. So now is the time to ask any questions to any speakers. If any questions came up, please raise the hand or use the chat function if there are any. We have a question in the audience.
Yeah, I can feedback that a minute.
Yeah.
I was wondering how the isolation works in Geneset of sample or data or whatever. So how does they get off the fluid of taking harm?
So this question is for you, Adriana. How do you get the fluid out of the brain as you showed the image, so that you cannot, so you can get the fluid out without any harm?
That's part of the treatment of the infants. So the clinicians do this process, not me, as a part of the treatment, and neurosurgeons put the device in the ventricles of those infants if they need regular tapping of the CSF. So we only get excess CSF that the clinicians and they don't need to. So that's a part of the clinical treatment. It's not been yes.
Okay, thank you. Did that answer the question?
Yes,
yes, thank you.
Thank you.
So there seem to be no further questions. So with that, I would really like to appreciate, first of all, the speakers that they were taking the time, and in particular, Andriana. I know it's very tight when you have to submit your thesis, so really appreciate you took the time to present, but also Dr. Borchett, Dr. Myringer, thank you so much for the great presentation. And if any questions come up, feel free to reach out and ask any questions that you might have. So you did see with these scientific presentations the bandwidth that is possible, like very different sample types, very different analysis in terms of microRNA and mRNA. And the beauty is really that you can work with really tiny material, as you saw for the CSF, but also very small biopsies from a tumor. A single section is sufficient where many technologies, other alternative technologies fail. And then also you have the bandwidth of the whole transcriptome and the melanoma, if you like, on many different sample types, and you're not losing any material or any markers with the extraction. But you can really be sure that you bring the assay to the sample rather than trying to get somehow the RNA out and then detect what is remaining after extraction. So here the team in Essen is really helpful and supportive for any projects you would like to run locally here. So we really also would like to support any discussions, in particular for your project of interest. Feel free to reach out to Dr. Myringer directly or to myself, and we will then discuss the details of your project needs in more detail. So with that, I'd like to thank all the attendees. Thank you so much for attending and for the great discussion, and wish you a great rest of the day. Thank you so much. Oh, there's one question, yes. Does the Institute of Pathology provide the mRNA-seq and microRNA-seq as a service for other labs?
So depending on what you want to ask, so if you need the RNA-seq with HTT technology or the spatial transcriptomics, we provide this also as a service for our labs. There's no problem. We don't do classic RNA sequencing. So we're not the ones for the classic transcriptome sequencing, that using sequencing technology. We are all on this histology, this slide-based sequencing format or sample sequencing format for the HTT, but not the standard RNA-seq or microRNA-seq. That's the question.
Yes, so in the case the question is related to what we saw today, so the microRNA analysis on mRNA in a target way with HGG, that's supported here locally. That's correct. Okay, thank you so much, everyone, and wish you a great rest of the day.